start lecturing, I get hungry. And all I can think about is that chocolate shake in the refrigerator. And then, oh yeah, I had biscotti for breakfast. That doesn't stay with you very long. That can include a silent mutation, it can include a missense mutation, or it can include a nonsense mutation. Okay, what if the mutagen, know what a mutagen is? A mutagen is a chemical or an agent that causes mutation.
Like reducing cigars?
Yeah. Those stripes in your charcoal broiled steak, the smoke in your cigar, those are partially uncombusted organics. And they cause something called benzpyrenes, which cause mutations, serious mutations. Those are one chemical. There's other kinds of chemicals. Some chemicals we purposefully give, and those are be used to fight viruses. Because viruses have to get inside host cells and be replicated, and if they're replicated with mistakes from the chemicals, then the virus is going to have all those mistakes. But we'll come to that later. Let's first stick with this. Mutagens. One is radiation. In fact, when I was an undergraduate, I worked with a certain mold. It was a black mold. And I hit it with radiation until I got a mutated strain of the mold that was albino or white. Because we were trying to see the differences in the pigment, what effect did the pigment have. Anyways, so a mutagen is a chemical that induces mutation. So some mutations are chemically induced. Some mutations are spontaneous. That means the natural occurrence. It's God's fault. I don't know. Spontaneous means it's just a natural mistake. But you can also produce mutations by hitting organisms with chemicals. If I hit the organism with a little bit of chemical, I might make a mutation happen in one spot. If I hit the organism with a lot of chemical, I can make the mutation happen in several spots, right? And the chemicals tend to be specific for the base letters. So there's a mutagen that will affect the C, so it's misread, and you get the wrong letter on this side. There's different chemicals that attach to the G, make it be misread, you get the wrong letter on this side, and so on down the line. Just want you to be aware that the more mutagen you expose your organism to, the more mutations you're going to get. Makes sense, right? Okay, there's another kind of a mutation. These are not spontaneous. The spontaneous mutations are always point mutations or base substitution mutations. Some mutagens can cause point mutations, or they can cause frame shift mutations. Here's a frame shift mutation. The chemical binds to the DNA and completely blocks the letter. And since your DNA is double-stranded, it would block both sides. But just think if it were trying to be copied and it didn't see the C there, it wouldn't put anything here, right? So when the DNA is being read, it reads C, C, doesn't see this one because the mutagen binds and covers it. And so it reads the first codon, CCG, reads the next codon, GGA, and the next one, AAT, and then TT, whatever, I don't know what I'm supposed to do there. So a frame shift mutation is the most fatal or the most harmful potentially. Potentially fatal because not only will it screw up the letter that it binds to, but it will screw up every codon after that because it shifts the reading frame of the gene. Get what I mean? Okay, now let's pretend my DNA is this long closed loop of DNA. All I could put on the board is one gene. Every gene has a sequence of letters that signals the start, and that's the promoter, and one sequence of letters that signals the end, and that's the terminator. This part is the functional part of the gene, or your book likes to just call it the functional gene. The functional gene is the gene that codes for a protein or some trait. But every gene has a start and a stop. And it's a good thing, because with frame shift mutations, if you shift the reading frame of the gene and there's no end to the gene, then that shift is going to occur throughout the entire DNA. But every gene has a stop, a terminator. This way, the frame shift mutation might completely screw up this gene. But at least it doesn't go to any other genes in the genome, okay? So terminator ends the gene and keeps the frame shift mutation from running throughout the DNA, yeah?
By terminator, do you mean like the stop codon, or?
Yeah, totally.
Or when that also misread?
It's a different thing.
It's like
In your DNA, the DNA has genes signified in it. What did you say?
I was saying that the terminator is the stop codon, and then also being misread.
Oh, the
by
codon.
frame shift.
That's a different thing. When you're reading, yes, when you're making mRNA from DNA, the mRNA will have start and stop. Because the DNA has promoter and terminator. Think of it that way.
Okay, so there's no start and stop on the actual DNA.
Right.
The start and stop are on the
RNA.
mRNA, or
Yeah, right. So when you're making a copy of a gene, you want to copy just that one gene. So the gene in the DNA has a promoter to say, okay, start copying now, and the terminator say, okay, you're done with the gene, stop now. And the mRNA is the one that has to have the start codons and the stop codons. Did that go along? Did anybody agree with that? She's like, I don't know anymore.
is it like the offline stuff?
Yeah, we're gonna do that too. Alrighty, yes, exactly what we're gonna do. But first we gotta reiterate. Okay, so, frame shift mutation, stopped by the terminator. What if the frame shift mutation didn't happen up here? What if it happened down here? Okay, so maybe I just screwed up the last codon in the DNA, in the gene, right? But if it's if the frame shift mutation is in the beginning of the gene, then you're gonna screw up the whole gene. But either way, the terminator stops it. It stays, the frame shift mutation stays in the gene. But what if the gene were a gene for an essential enzyme? An enzyme to run your Krebs cycle, perhaps. An enzyme to run your glycolysis, perhaps. Then it could be fatal to the organism, or the cell, whichever way you want to think of it, because with bacteria, the cell is the organism, right? And that's why they study operon theory in bacteria, because they're a lot simpler organisms. Okay, so you understand your different types of mutations, and the degeneracy of the genetic code, and what a mutagen is, and now we're going to do this over here. And then we'll do induction and repression. Okay. Now, protein synthesis steps. This is the one thing I didn't do, was do the steps with you. I kind of went through the process without telling you the names of the steps. So let's put a big bacterial cell up here. Plasma membrane, cell wall, nucleoid. Let's do our flower-powered nucleoid. We're going to open up part of our nucleoid. because we're gonna make mRNA. So this is your double-stranded DNA, right? You've got your little hydrogen bonds. Not very good at drawing my dotted lines. Okay, the hydrogen bonds temporarily break. mRNA is copied from one side of the DNA. Here would be your promoter region and your terminator region. On your mRNA, there would be a start and a stop. And the functional gene codons would be in the middle, if that helps at all. When the mRNA is done being copied, it literally pops out, leaves the nucleoid, and goes out to the cytoplasm, where it's translated on a ribosome. And as it's translated on a ribosome, you end up getting a chain of amino acids. Right? As your mRNA is read, you get a chain of amino acids, and it twists and bends and folds into a functional protein. Okay? The first step, protein synthesis steps, actually there's an initiation step. I won't ask about initiation. Just means when it's being copied, the mRNA is being copied at the DNA, it has an area that says, okay, let's go. Let's start the process of protein synthesis. It's a signal to the cell. First step is transcription. Transcription happens at the nucleoid. Cell makes a copy.